Hi guys, in this video I am going to cover all the essential points you need to know about glycolysis to master your board exams, most importantly talking about USMLE Step 1. I'm gonna try to make it so simple for you with focusing on the most clinically relevant points only. So please subscribe, let me know your opinions and leave me your comments when you have finished watching this video. So what's glycolysis about? What's the goal and the end point of glycolysis? Glycolysis is the most basic pathway of energy metabolism within the cell. In glycolysis, glucose is converted to two molecules of pyruvate, and then pyruvate can enter either aerobic metabolism or anaerobic metabolism via conversion to lactate. I'm going to talk about the details of this in another video. So, the final goal is to generate ATP, which is the energy currency of the cells. The glycolysis itself produces a tiny amount of ATP, and I'm going to talk a little bit about the importance of that later in this video. So, first of all, glycolysis happens entirely inside cytosol. And there are several steps in glycolysis. You don't need to know the name of enzymes for each and every step. And here I have highlighted the key enzymes which are essential to know. So let's start. What happens when glucose is entered into the cell? And an enzyme adds a phosphate group to glucose to trap it inside the cell. We will call this enzyme a glucokinase or a hexokinase, and there are differences between these two enzymes. I will talk about that at the end of this video. So let's just quickly say that we have two types of enzymes that add a phosphate group to a substrate. And if ATP is used, the enzyme is called kinase. If no ATP is used, we will call it a phosphorylase. So now here we have a glucose converted to glucose 6-phosphate by a kinase enzyme. In the next step, glucose 6-phosphate is converted to fructose 6-phosphate, and then fructose 6-phosphate is converted to fructose 1,6-bisphosphate by the enzyme phosphofructokinase 1, PFK1. PFK1 is the rate-limiting enzyme of glycolysis, and it's heavily regulated. So this enzyme is induced by AMP and inhibited by ATP. So when we don't have enough ATP, we don't have energy, we want this enzyme around. And when we have a lot of ATP, we don't want it around. Additionally, citrate, which is one of the most important substrates of TCA cycle, also inhibits this step. When we have too much citrate around, we don't want glycolysis to run. Okay, so let's get a closer look at how this enzyme works in close relation to insulin and glucagon. So here we have fructose 6-phosphate converted to fructose 1,6-bisphosphate. Alternatively, fructose 6-phosphate can be converted to fructose 2,6-bisphosphate by the enzyme phosphofructokinase 2, PFK2. And then fructose 2,6-bisphosphate enhances the activity of PFK1. Insulin and glucagon act in this step right here. So insulin induces PFK2 and glucagon inhibits it. So indirectly, insulin enhances glycolysis and glucagon inhibits it. So let's jump back to the cycle. In the next step, fructose 1,6-bisphosphate breaks into dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. And dihydroxyacetone phosphate, DHAP, can be converted to glyceraldehyde 3-phosphate. So for each molecule of glucose, we have one DHP and one glyceraldehyde 3-phosphate, and DHAP can be converted to glyceraldehyde 3-phosphate. Therefore, for each molecule of glucose, we have two glyceraldehyde 3-phosphate, and then two molecule of pyruvate at the end. Okay, so let's talk a little bit about these ATPs in here. In these two steps, the kinase step and the phosphofructokinase 1 step, 2 ATP is consumed. And then here and here, ATP is produced. And remember that we have 2 glyceraldehyde 3-phosphate for each molecule of glucose. Therefore, 4 ATP is produced and 2 ATP is consumed. Net ATP production would be 2 per glucose molecule in glycolysis. Let's just say that this NAD in here is converted to NADH, and NADH can be later used in oxidative phosphorylation in cells that have mitochondria to generate additional ATPs. But if the cell doesn't have mitochondria, then it's heavily dependent on these 2 ATPs coming out of glycolysis, such as RBC. RBC doesn't have a mitochondria, and so it's heavily dependent on the anaerobic metabolism and on the 2 ATP molecules coming out of glycolysis. So let's jump back here. Glyceraldehyde 3-phosphate is converted to 1,3-bisphosphoglycerate, 1,3-BPG. And then here we need an NAD to pass this step. So we need to regenerate NAD to run the cycle. Where is this NAD regenerated? So if there is an aerobic metabolism, it can be regenerated in oxidative phosphorylation in mitochondria. Otherwise, it needs to be regenerated when pyruvate is converted to lactate. And we will need NAD to be here to run the cycle. So moving forward, 1,3-BPG is converted to 3-phosphoglycerate. And here is one of the steps that produces ATP. Alternatively, 1,3-BPG can be converted to 2,3-BPG. And do you remember where does this happen? Yes, it happens in RBC. So if RBC is in the oxygen unloading state, the concentration of 2,3-BPG should be increased, which means that 1,3-BPG will be converted to 2,3-BPG instead of a 3-phosphoglycerate, and it will be in the net expense of this ATP in here. And guess what? RBC is heavily dependent on this glycolytic ATP, and it has to sacrifice this tiny amount of ATP it has to produce 2,3-bisphosphoglycerate. Interesting. So as we go to the bottom of the cycle, we arrive at PEP phosphoenol pyruvate, which is then converted to pyruvate by the enzyme pyruvate kinase. This is another step that produces ATP, and it's an important one indeed. So what happens when we have pyruvate kinase deficiency? It causes a form of hemolytic anemia because RBC won't have enough ATP and therefore no energy to keep the NAK ATPase pumped, and so we will have hemolysis. And also in this situation there will be an accumulation of 2,3-BPG upstream inside RBC. So keep in mind, pyruvate kinase deficiency is associated with reduced ATP, increased 2,3-BPG and hemolytic anemia. And finally, let's wrap up this subject. As we learned earlier, we have 2 types of enzymes to initiate this cycle, glucokinase and hexokinase. So let's see the differences. Glucokinase is found in liver and pancreas. It has high Km, meaning low affinity for glucose, and high Vmax meaning high capacity. In the other hand, hexokinase is found in other tissues. It has low Km and low Vmax, meaning high affinity and low capacity for glucose. And then glucokinase is responsible for maintaining blood glucose, while a hexokinase maintains intracellular glucose. And glucokinase is the one which is induced by insulin. So what happens when we have hexokinase deficiency? Again, RBC is heavily dependent on glycolysis, so we expect to have hemolytic anemia. And when there is a glucokinase deficiency, insulin secretion will be impaired, and patients will have a type of congenital diabetes called MODY, maturity onset diabetes of young, which presents with early onset, non-progressive diabetes, i.e. patients have high basal glucose levels without any symptoms of diabetes or without developing diabetes-related complications. So this brings me to the end of this video. Please subscribe to this channel if you like this video and give me your honest opinions. I'll continue this if I get enough positive feedbacks from you guys. Thank you.